Increasing temperatures are pushing potato farmers higher up the Peruvian Andes. But what if, rather than trying to escape climate change, they confront it? They need to plant potatoes that are more resistant to threats. But those super potatoes aren't readily available. Fortunately, some wild cousins of crops have traits and characteristics that their domesticated relatives have lost. Pre-breeders can combine the best features of different potatoes and get those traits back into the cultivated gene pool. To visualize pre-breeding, think about breeding a house cat with a tiger. You may want the strength and stamina of a tiger combined with the size and docility of a cat. But you won't get all that at once, so you keep breeding your initial offspring with house cats until you get a really strong cat which still purrs. This pre-breeding forms a bridge between the gene banks who safeguard the wild relatives and the plant breeders who use them. The Crop Wild Relatives Project is now supporting a global pre-breeding initiative that is bringing back the diversity in 19 important crops. All of the plant material and data generated during these pre-breeding projects are available in accordance with global agreements, so plant breeders anywhere in the world can access them in ready-to-use form. If farmers in the Andes and around the world go back to the place of their initial cultivated lands, despite higher temperatures and new diseases and pests, they'll be thankful there's a bit of the wildcat in them.
Greetings, everyone. On behalf of the Gene Bank Platform and the Crop Trust, thank you for joining us today. I'm Yanni Van Beem, and with me is Faye Wambua Ludeling, who is co-moderator for this presentation. It is my pleasure to introduce our speaker today, Dr. Filippo Bassi. He holds a PhD in plant breeding and genetics from North Dakota State University in Fargo, United States, and a master's degree in plant biotechnology from the University of Reggio Emilia in Italy and the University of Adelaide in Australia. Filippo is a senior scientist in ICARDA's durum wheat breeding program, which aims to deliver superior varieties capable of withstanding the whims of climate change. He was the recipient of the 2017 Olam Prize for Innovation in Food Security for the release of a heat-tolerant durum wheat variety adapted to the conditions of the Senegal River Basin. He is a member of the Wheat Initiative Scientific Steering Committee, which oversees the expert working group in durum wheat. In 2020, he was also awarded the second prize of the Ted Crosby Innovators. To date, his efforts have led to the release of some 34 varieties in 16 developing countries. He deploys the most advanced breeding techniques to try and pack into seeds of new varieties some critical traits for climate change adaptation, often starting from wild materials obtained from the gene banks. This month's Grow title is Squeezing the Good Stuff Out of the Vaults, Fields, Genomics, or Gene Bankomics. Welcome, Filippo.
Thank you very much, and thank you to the Gene Bank platform for giving this opportunity to present some of the things that we do here at ICARDA in an optic of trying to really have a conversation with all of you. I mean, I was looking a little bit of the participants as they were coming up, and I see so many friends out there, so I want to wave everybody and hello. It's a shame we don't get to see each other more, but and really, I don't know what came into me when I came up with this title. It was I was on holiday, and I liked the idea of having something strange. I take the advantage, of course, of thanking also all the many co-authors and colleagues that have contributed in a way or another to this presentation I will be giving, and the many donors that, of course, are contributing, including, of course, among one of the most one, which is the Crop Trust in North. I like to start with this image to see if you can all come much closer to the screen and try to have a look at what it says here. These are the pedigrees of a field book that is actually our international nursery. So it's the material that we provide every year to more than 50 partners in some 40 countries around the world. So it's the most advanced and best germplasm each year that reaches this status. And underlined in red, I think you can read Triticum agelopis crosses, you can read Polonicum, you can see Urato in there. In fact, my finger is pointing exactly at the agelopis crosses. It's a simple cross, and you see how good that plant actually looks. So, and this one next to it would be Polonicum and so forth. So I don't know how familiar are you with durum wheat, but I assure you these were actually very good lines that performed very well. So this just gives an idea of how much Picarda, at least in my program, we believe in this type of genotype. If you don't mind a little bit of conversation, I don't know how many of you are familiar with Picarda. We are a CG center, and our goal is to work from the non-tropical dry land of the developing world. They're represented basically by the diagonal white lines you see on this map. We have office spread across a little bit of the target region, which is quite vast in reality. And in red, with the arrows, I'm indicating our main research station. One is in Morocco, the other one is in Lebanon, then in India, and Syria that we're still operating. And with the star, you actually notice where our gene banks are located. So 3 of our gene banks actually use exactly the same field as myself. So that shows already the type of interaction. Then we partner with our national counterpart, our national partners. We have several operated stations by them, including maybe the most important one among them is the drought platform in Morocco, is the RAS Center in Turkey, 2 very important heat tolerance sites in Senegal and Sudan. But of course, there's also Ethiopia for RAS and many others. So to go back to how much in wide process we trust, to date, the program has released 143 recorded varieties. I contribute only to some toward the end. In 22 countries, and when you look at it, they're represented in this graph here by the orange bars would be which one of them were actually derived from land races crosses, and in blue, which one were derived from crop wild relative. About 10% of that total number is crop wild relative derived, and 29% from land. What is maybe more interesting is that if you look in the past 5 years, or much more recently, you see the number from 10% went up to 28% for crop wild relative and to 38% from land races. So there is a trend toward actually making more of this material become actually a variety, so having an impact on farmers' field. And I want to show it a little bit this way, how to make things work. Well, first of all, I'm not a Barcelona fan. And in reality, I also take the opportunity to send this little dart to those fans that this incredible striking formation is no longer part of the team. But once upon a time, they were. You had Messi, you had Neymar, and you had Suarez. And I think it's really very similar to what I can think of in a breeding gene bank continuum, where you have Messi, that is your gene bank or gene banker, if you really want to keep the same naming, that creates all that diversity, all that dribbling and so forth, that passes on to Neymar, which is the pre-breeder that brings much more closer to the goal, and then with that final pass and final assist, the breeder comes in, that's Suarez, that scores the goal. When all these things is a linear system, things happen and happen fast and happen well. When that is not a linear system, then things do change. I took advantage of this beautiful slide, of this beautiful image created by Albert Schutzles of IPK as part of an article that we have recently submitted. Hopefully will become a full article. It looks at what breeders really look into the gene bank collections. So one way the breeder look at it is a qualitative transfer. The breeder wants one trait and nothing else, just wants that one. So that one has to be identified. So we will have to do a characterization of our plant genetic resources. Then we will rather do a back cross, like in this graph, or as we do mostly at the Picarda top cross, to try to get rid of as much of that unwanted diversity as possible. Many people suggest the use of marker selection, kind of follow more quickly the traits that you're introgressing. Of course, that does require an extra investment when you try to make it, to identify the marker first. And in reality, if the elite that you're using in your recurrent system is good enough, was really selected to be among the best at the time you start the process, and your process is not overly long, then often enough, what you end up is a great line with an additional useful traits. So that typically, in my opinion as a breeder, that is ready for direct. It's not a line that should be used for crossing. And sometimes I call it the fear of the pre-breeders. They're always afraid of talking about releases because it seems that it's not in the job description. So they have a great line that will tell you, look at my data, I have a line that yields better than the normal elite, that has better quality than the normal elite, that has better disease resistance than normal elite. And I use that as a crossing parent. And it's like, you know, if it was a breeder saying that, it would be a direct relate. So I just want to point that out, that if you select your elite well, we go all the way through. On the other hand, for a long time, it's called the pre-breeder, or maybe the cytogeneticist would be even more correct word, tended to use genetic stocks that were not very performing, like, you know, in case of wheat would be Chinese Spring or Pata or Langdon. So the final results was indeed a transfer of that trait, but then in such a bad background that the breeder still had to do a lot of work. So I think that step of interaction of selecting the right material to transfer to, it's very critical. In fact, to the point that we came up with some sort of a scheme as part of this submission. To me, everything starts down here. And just to make it clear, I talk about breeding and pre-breeding pipelines. But in our case, in our program, most of it is actually the gene bank that does the initial pre-breeding, and it's my program that takes it up to the end. So in reality, there's not a real separate person doing the pre-breeding. But I want to call it as a pre-breeding. So it starts here, and it's actually incredibly important, and it's surprising how often that does not really happen. A breeder needs to define the goals in terms of what are the traits for which I need an investment, for which I need to find new diversity. I've seen in some cases, pre-breeders coming back to me and say, oh, I have this great line for salinity tolerant, but maybe I don't work on that trait. So, this is important to discuss it first and define those traits together. Once that is done, then most of the work really happens here, which is defining the fraction of that diversity that is the most useful for those traits. And of course, the gene bank have a huge role to play here, and the collection of the main primary source. Another thing that doesn't happen quite enough, I feel, is this continuing interaction between breeders and pre-breeders. As breeders, we try every year to have a genetic gain, which means the line that they gave last year to the gene bank are somewhat already old compared to the line that I developed this year. And that type of continuous improvement has to enter the pre-breeding pipeline. Otherwise, somewhere we are making an advance, but at the same time, we are falling short. So every year, the breeder has to define its champions and has to pass them to the gene bank or to the pre-breeder for the hybridization. At which point we come to the newer technology where we really try to accelerate the fixing of these alleles, rather than breeding, marker seed selection, genomic selection. There's a series of them. And then there are two alternative pipelines. One is at this point, the pre-breeders have developed new parents. They come into the crossing block of the breeders that can finally use them. So we have a champion with a plus that has become better than what started. Definitely didn't look like the Langdon sac here that was growing almost 2 meters tall in a glass house last time I was in Fargo. Another principle that we use a lot at ICARD actually is that F2 or F4 or still segregated material passed on directly to the breeders, that then takes it from that point onward and try even to release a variety directly. So this is a little bit of the things. I just want to give another example of what it really means. So on one side, this is our gene bank. You see, when you find a good line, how many crosses they have to make to it and how challenging that is. That is, of course, Hamed Amri, that I need to thank always very deeply for what we do here. And they have a lot of diversity, of course, in the field. So they start screening. For instance, they were looking at different traits, rooting capacity, spike fertility, leaf rust resistance. They looked at into the Dicocoides, Uratum, Boeticum, germoplasm, and they find, for instance, that a dicocoid had large grain size that could be used. So if you were to do this alone, then all these traits are important, all these material is important, and all of them need to be brought into modern material. But if you do it together with the breeder, there is a change. First of all, there's a breeding priority, right? So if you look at my own breeding program, Septoria tristis is not a major focus on my program. Yellow rust is not a major issue of the program at the moment. So we say, okay, don't make these crosses because I don't care. And also we can look how difficult it would be for me to take whatever they developed and bring it to something final. So again, working together, basically I can reduce significantly the workload of the gene banks and really focus on what it would be the most useful. So that type of interaction is so important and needs to happen, to me, very, very early in the pre-breeding cycle. One example, maybe one that most of you are familiar, I'm not sure, is Asian fly. Asian fly is a very nasty pest, it's down here. A resistant variety is perfect, a susceptible becomes basically dead. It's calculated in a lot of countries that are suffering for delayed delayed on-season rain, that the yield losses can be very significant, about 30%. So it's a major focus of my program. In 1989, a group of students from Morocco went to Kansas State University to work with the team of Bikram Gill, and together they identified 4 accessions of Triticum araraticum that were actually resistant to Asian fly. They brought those back from Morocco and they worked on it for a while. And 14 years later, in 2003, they were able to obtain the first release of an Asian fly resistant cultivars from Morocco. These had a lot of shortfalls, so it took 4 more years. In fact, there were basically 3 top crosses extra. You see here, Araraticum by Arthur was a back cross, the original one, and then 1 top cross, a second one, and a third one, to come up with a new variety that's called Farage, that actually is now taking up some area here and in other places. So, you know, when you look at 2016, this is a part of the country of Morocco. The color red means that in February, things in terms of agriculture were going pretty bad. And by 2016, it becomes violet, which means that we're actually plowed in or grazed directly by animals. So the drought was so severe that it was pretty catastrophic for the country. And that year we had 2 varieties, the most commonly grown, and this Farage resistant to Araraticum planted side by side in the same size. And you can really see the difference between the 2 in terms of productivity. And since I still have a biotechnology background, I want to see if I could do a little bit more and have a little contribution to this long story of Asian fly resistance here. So in 2019, we were able to publish, so 30 years after the original identification, a good marker for following this Araraticum gene or segment for Asian fly resistance. And you can see that it predicts the resistance with 100% value. So everything that is declared resistant is really resistant. Then there's a little bit of false negative, but all in all, I can now take this and really push it through the breeding program. Just to show you 30 years to come to a point where you can really scale it into the breeding program. Clearly, it is a success story, but one that I hope in the future can be told in maybe 5 years or 10. So there is a learning experience here. In fact, we think we learn. With the expert working group in Durum Wheat, under the Wheat Initiative, we have launched this global Durum initiative called the Global Durum Panel, where we receive more than 3,700 entries, modern land races. You see, there's tetraploids, there's land races, there's different modern breeding program that should represent somewhat the global use of durum wheat in terms of breeding and rebreeding. All came together, and then we select the best one. And once we did, we genotype everything, and now we have provided back to more than 45. Actually, today I count, we have 50 partners working on this, and each one of us is doing whatever phenotyping can be done. So the idea is that, you know, tomorrow in Ethiopia is discovered that one of the line is very resistant to stem rust. The person that does it would yell to the whole community, look, use this one, and then every breeders around the world can take this one line and cross it to everything and have basically a global crossing block. On top of that, you can immediately run association mapping and even come up with a marker, because maybe I'm sitting in Canada, I don't have stem rust, but I'm worried that it will come. So I can use the marker to make sure that my germplasm has whatever good thing you have developed there. So the idea is that by doing this as a community, we should shrink down the time from 30 years that we did for Asian fly to almost 5 years to really move things quite quickly. And as we were doing that, of course, we studied a little bit the diversity that the exploitable durum wheat diversity had. Of course, there's much more than this in the gene bank, but this is what people have been using for the past 10 to 15 years. And then when you look at this phylogenetic tree here, it looks a little bit what most people might expect. So you have on the left-hand side, you have, let's call them the tetraploids, the ancestral parents, durum wheat. You have on the right, all the durum land races divided in different clades. And basically down here, you have all the elite modern germplasm that, it's probably in farmers' field somewhere or should be soon. And when you look at it, basically, these are the funders. Only this part of diversity was the funder of all that diversity. There, it means the part on the right and the part on the left was not used by breeder to develop modern plasma. So clearly, there is a huge amount of genetic diversity that's not been reached. However, when we went and looked a little bit more closer, the first 4 are actually land races, and then everything else at the bottom is modern material. We look at minor allele frequency, which is typically what you look in your diversity is a good minor allele that is lost somewhere. And you see the program like ICARD, I have more than 50% of the global available minor allele captured in the program, which is quite significant. And is, as a comparison, the landrace from the Mediterranean, 65% of them. So you're really not that far. And then when you look at a program like the one of CIMMYT, very good at actually capturing very rare allele. Things are very, very unique. And they have 20% of them against what landrace have only 6. So there's 2 learning curve for me here. The first one that we should give more credits to breeders. They are actually handling much more genetic diversity than one would normally give credit for. Of course, this is the case of durum wheat. I don't know for other crops, but for us, there's a lot of diversity that is actually within the breeding program. On the other hand, diversity per se is not strictly useful, really. We don't want all the diversities here. What we want is the useful part of this diversity that should be brought. So there's a good element for both. My compliments to the breeders, especially the one of the beginning of the century that really did great work in keeping genetic diversity wide, and the potential to bring new ones from different sources. So looking at it, I would just want to put this picture here. This is actually part of the panel that I was talking about, but this is the part of the land races. So as we were walking through with our colleagues, and it's Pablo Roncallo from Algeria, and this is Rukus, one of the guys that really took care of business in Lebanon, we came across this land race here from Ethiopia that was completely devastated by yellow rust. Right? So normally we just keep on walking. Instead, we stop in front of it, start discussing, yes, this is yellow rust, but there's a very long spike. It's very early, shorter than the rest. So maybe there is something useful here. Maybe 10 minutes after in conversation, we kept on walking, we did not make crosses to that one. But that type of uncertainty is actually what really is causing what they call immobilism by overabundance. If I take took yesterday the overall Genesis database, just looking at Triticum turgidum land races and wild material, I was indicated 46,000 accessions are available for it. That's a lot. Okay, you see them spread out here, including this very, very interesting, I think the gene bank community calls them the salt tolerant land races. And I just out of curiosity, okay, well, how long would it take me to do a cross to all of them? And my program, if alone we just try to do a cross to each one of these accessions, it will take me 50 years, which is actually less than I thought it would take me, but it means I would do nothing else but that. Anyway, most pre-breeder, of course, breeders ask, what if I want to use, what should I use? And the answer to that question sometimes is much more complex than one would imagine. It's not so linear. Okay, tell me what to use and I use it. It's quite complex. I find a good solution that the colleagues at ICARD have developed is this focus identification of germplasm sources, so FIGs. The idea here is that you start with the passport information with the GPS location where a certain landrace was collected or wild relative. You add to that a series of information like, which elevation was collected, what was the climate, the soil. Then you work with colleagues, physiologists or pathologists in this case. You make a model to predict whether or not yellow rust, for instance, could occur at those places based on the climatic condition. Then you create a probability score and you turn out that, these many accessions here to the right-hand side, the green one, have a high probability of being resistant based on the information of the place of collection, on the passport. And then you get these results. So you select only 293 sessions that are likely to have success instead of the thousands that we started with. About half of them were in fact confirmed to be resistant to that. And as you go across to different diseases, or even drought actually works exceptionally well, you can actually get quite a bit. So this is a good way of simplifying that question, what should I use? With my colleagues, and I hope you can read the link here, but it's the same that you have down there. Under the leadership of IPK in Stein, we have started a project funded by the Horizon 2020. It's called Agent Activate Gene Bank Network. And what we are trying to do is working with 15 gene banks, try to deploy genomic models to even better predict what these land races would provide. So for instance, at ICARDA, we would do a testing of some material for drought tolerance. Everything is genotyped. And then we can try to predict within the IPK collection, which one of their land races is actually also drought tolerant. So this actually works surprisingly well. This on top here, this is a nice paper by Zaccaria and other colleagues here at ICARDA. They were trying to predict kind of in this way, the grain characteristics, the seed characteristics, so the area, the perimeter, the length, and the width. And here you have the accuracy. So you can see that really with good accuracy, over 70% or 66%, it could actually predict what a land race would look like in terms of grain length on the basis of only few being phenotyped. So that's quite well. And now we're even going beyond. We're adding to this prediction the climatic models, and we can see, so we call it FIGS plus, so genomic plus climate model, and we see that our prediction go even. So this is really a nice future trend for the generations to come. But, for diseases and for simple traits that have low G/E effect or high irritability, that's okay. But this is, for instance, is a testing that I had in my station back, I think, in 2015. The upfront here, you have land races. So clearly that one is very susceptible to yellow rust. You might not use it. And in the back, you have the modern material. So if I start looking at the yield performance, because never forget that most breeders, their job tends to be deliver a continuous increase in their yields, in the genetic gain for yield. And every time you work with material that has lower yields, you're afraid that you will not be able to do that problem. So of course, when you compare, and this was a very dry year where land races were actually were advantaged. This is the yield, and you see the performance. So you have some top modern material here. This is the top land race. This is the average. So a not so silly question would be, if you had a top yielding land races as a breeder, would you use it? Again, it's not a simple question, because really, is that what you're looking for in land race? It's a valid question. So it talks about quantitative transfer. In this case, we're no longer looking for a single trait. We're looking for the collection, the value of the collection. We make a cross. We develop PGR by elite cross. And what we get is a distribution where ideally you want to be here. Better than the elite, you are improved for that trait. We see the three-way crosses are the best. And the problem is that all combination of plant genetic resources by elite are extremely challenging to predict on the basis of the available genetic value. So a good land race by a good elite does not give you a best hybrid. A bad land race by a good elite could give you a best hybrid. So there is a big complexity here to be captured. Again, thanks to Albert for this beautiful image. One example that I want to share is the case of a variety of machine. It is a top cross between Triticum dicocoides collected from Syria, Amedacul, which is an elite of ICARD, and Lucus, which is another elite. The cross was made by the gene bank on the simple fact that Triticum dicocoides was an interesting source of diversity. So there was no specific traits in it that we want to capture. Then the gene bank passed the F2 F2 material to the breeders that selected it as if it was its own line. In 2017, this line was released under the name of the sheet. It has much larger grain size. It performed an astonishingly well in dry conditions, and surprising enough, also when we put in irrigated condition was one of the top yielders. Definitely not something that we knew when we made the cross. We had no idea what the Coides brought. To really show that we had no idea what we were doing, we went beyond any reasonable doubts and actually turned out that this variety is greatly performing in the Sahara of Morocco with using drip irrigation and now providing great couscous for this female cooperative. So really, the cross was made without knowing all these things, and the results are great. As a scientist, we want to know why that was. So we went studying and we realized the roots would be the reason. In fact, this sheet has a very deep root system that is capable of capturing any moisture left in the soil, and that's actually gives very good yield advantage under dry conditions. And we came up, realized there's basically three major QTLs that account for this, two of which comes from the Dicocoides parents, and together they account for more than 300 kilograms per hectare under dry condition and 39% of the overall. So that cross that had no idea where it was made went all the way through. Now we know why that Dicocoides was useful, but that didn't stop us from using it. And now we are much better positioned into taking this allele and really moving through to the breeding process. And that paper was considered interesting enough that received the 2020 Outstanding Paper Award by Crop Science. So the reference is down here. If you're curious, it's a simple paper, but I think it's fun. We expanded that study, doing comparing crop wild relative that are represented here by the orange boxes with the cultivars and elite across 20 environments in Morocco, Lebanon, Ethiopia, and Senegal. See the environments here being grouped based on their major climatic conditions. And here you have the grain yield. You basically see that the environments 1 and 2, the crop wild relative were actually the top performance, and as well in environment 4. And just out of curiosity, environment 1 and 2 did not have any evident climatic stress. You can actually see the yields are quite high, while environment 4 was dry conditions. So again, these crosses really kind of working well in the program, to the point that the crop trust and NORAD lived in us to go beyond the station into farmers' field. We have a project called DIVA, which is about participatory research, looking at the whole value chain with farmers and female cooperatives. And this is kind of a summary results of that. There were 25 farms trial conducted in 2 very dry seasons, 1819 and 1920. I present them here divided by rural communities. And here there's the yield advantage of the best crop wild relative compared to the commercial line the farmers were using before. You see that in some cases the number are huge. Actually here there's quite a bit of Asian fly involved. That's why really the yield jumped up. Also the difference in value is quite small. The yields are small. But since I heard that before, I also want to show that this does not happen only in the developing world. We send every year material to Australia. It's tested in what is called the CAGE program. And here on the left, you have the major check of Australia, Hiperno. You see here is a commercial variety that's promoted. Here you have different station with the actual average yield, and it's presented as a CVE. And here you have the Carda material, and you see that in the case of land races, here North Star and Dicocoides derived material, they were highly performant, better than the check. Same here, same there, same here, and same there. So that this material not only performed in the developing world under dry conditions, but it performed also in the developed world under good yielding potential. So it's really working well. There is, however, somewhere linkage drag that we have read so much in the books, and that linkage drag actually in our own material, we identified it in the quality. You can see that when you look at sedimentation index, which is an important factor for durum wheat, and the color flower, which is also an important factor, the cultivar was so much better than the crop wild variety. And that is actually simply because we never thought that this crop wild relative process would make it directly to varieties. We always thought that they would become parents. And because of that, we had not done enough quality selection in them. And now that we have started doing it, things are really changing, and we see quite a few of these wild relative derived material becoming also top quality. One more example, this is the case of Senegal. So I'm becoming a Scandinavian program. One side I'm supported by Norway, the other side I'm supported by the Swedish Research Council in SLU with Professor Ortiz. And in this project, we wanted to see if we could add one crop along the river, because here there's two cultivation of rice, one is irrigated, one is rainfed. And then there's a fallow period that is rather short, but the temperature are on average up to 32, 33 degrees, but in reality, it can be much higher. So we have tried to develop, actually succeeded in developing durum wheat variety that would fit in that cycle and perform quite well, well above 3 tons per hectare. And these are the two releases. One is Habi, which again has a silicondicocoides in it, and the other one is Amina, which again has speltoides. So we had modern material, but simply did not make it quite as well as these crosses with wild. And the story spread thanks to this bestowing of the Holland Prize to the whole team. I received it, but it's the whole team that was awarded. And now, you know, these are being tested by farmers across different countries in West and in South Africa. And you see some cases, the results are not great, in others are pretty good. So things are moving, and we're very pleased. Again, we did not know why those crosses were made in the first place. And then we went in, we want to study it. So we did this study using a plastic tunnel. We placed them at the time of flowering. That increases the temperature inside by 10 degrees, so important increase. And you see the difference between a line that is inside and the same one that is outside. When we look at the results, this is grain yield. You see how in the normal condition was almost double, and under the heat stress, the plastic tunnel. And we realized that the major factor changing was actually the grain number spike. So the fertility of that spike was heavily affected by heat, probably, you know, desiccating the pollen or affecting the ovaries. And when we went to look in the molecular component of it, we realized that actually 2 QTLs were controlling it, both of which were coming from speltoides. And together, these 2 QTL amount increased by almost half a ton the yield and the severe heat stress. You can see here, you know, 2 positive QTL, the 2 positive allele, and the grain yield on average. And on top, this is a combination of 3 heat stress environments, right? And this is when you have only one, and this when you have none. Again, this can be also. So I think I gave you some example to give some value to the words that I'm going to say now, which is, I guess, the reason that you know, we want to have this conversation with all of you about, you know, gene bank and breeders. And to me, this is how I converse with my gene bank. So every year we'd sit together. Actually, we will be doing it next week, for instance. And we designed together 20 top crosses. So it means A by crop quality by B, using the best elite I have, and the most interesting crop wide relative that the gene bank has to offer. The gene bank takes care of the most difficult part, which is to produce viable F1. Of course, there's embryo rescue involved, there's fertility, the restoration, there's a series of things that are involved there. They plant their F2, and I come through their plots. I select 30 spikes for myself, and these go through the normal breeding pipeline, now including also quality testing that had not. And on the other hand, the gene bank has a different responsibility than me. So I have a license to kill. I can remove material if I want. I don't have that license. So they need to maintain everything, bring it to inbreeding, and then try to screen them for as many traits as you can. So if I miss anything, they can give it back. And just 2 examples here. And by the way, the crosses to land races, Ricardo, we do it as breeding crosses. I can do them myself, so I take care of those myself, so that the gene bank is free to work on the compiler. This is an example. So you have 2, 3, 4 sister line. All the 4 came actually from the same cross with a wild relative. So these 2 looks incredibly promising. They are very, very good. These 2, the breeding pipeline failed to select for the right phenology and height. So happens, but I feel that the results are worth the cost of bringing this forward. Same here. Great sister line from a landrace cross, and one that remained way too tall and should have been selected better. And as we learn how to do this selection, my technicians as well and everybody, we hope to become even more effective in doing it. I'm sharing now with you the stage 1. So it's the first EL trial we conducted at Carda. There are more than 3,000 accessions involved in it. And I divided them by the gene bank made crop wild relative crosses, which means I selected them into 2, the landrace derived, which I do myself, and what I would call a normal elite by elite cross. And you see, of course, that the elite by elite, we have a much bigger number, 2,700 of those progenies coming from about 600 crosses. This is the column here, and they tend to be the top yielding. You see, these are 2 seasons, one high yielding, one very dry season. You can see in both conditions, the very top line was in fact an elite by elite cross. The land races, we do about 50, and we bring forward about 300 progenies to this level. They tend to have more crappy materials. The one that I was showing that was lodging would be this material here. And then you have the wild relative. In this case, we were better in selection. And again, they were the second best, but not the best. And the wild relative comes from 20 crosses, 120. However, what we call success rate is how many of these get moved to the next phase. So to stage 2 means they were good enough that can be moved forward based on quality, on diseases. And the crazy thing is that when you look at stage 2, only 30 progenies of the wild relative made it to stage 2, but this came from 15 crosses, which means 25% of selection here and 75% of retention of the original crosses. So that's quite successful. Landrace is not quite so good. And this is the elite where you have 200 crosses, about 1/3 only make it stage 2, and 440 progenies, which on one side means that probably need to do better crosses, but still. The question is why? Breeders, when they ask why, they look at this at the genetic gain equation. You have different function here. The first one is accuracy, how good are you in predicting? But in reality, in selecting, in reality, both the wild relative material and the normal are under the same condition. The years per cycle is very important, but again, the pre-breeding pipeline tends to slow down the breeding program, so it's not very good. Heritability, the traits is the same. There is maybe a little bit easier capacity to discard some material, but again, doesn't seem to be a factor. In fact, what we think might be the reason why only 20 crosses of wild relative make it all the way to the end with a much higher success rate is intensity. Number of crosses is lower, but the amount of alleles they're segregating is so much higher that you can find so much segregation and so much potential to find the best. So this is an hypothesis that we want to study whether or not intensity. I was asked to give my 4 cents on what I can see the gene bank of tomorrow be. I think the role for me, the gene banks to guide the breeding and the pre-breeding remains so critical. I mean, walking the field with people like Ahmed Amri that tells you, this land race came from an oasis in Mauritania. If you're looking for intolerance, I'm sure it has it. That value to me would be much higher than if he had characterized it for, say, disease resistance. Or the fact that it tells me he was collecting in a crazy place and could really give me something, to me there's no way of giving a price to that value. There's a potential to improve the way we characterize our land races using genomic models. That's interesting. I think it's a good way to give some information on major traits. But on the other hand, I always ask myself, how many disease-resistant genes do we need? The fact that the plant genetic resources are easier to be screened for disease resistance makes it the first trait that people look for into them. But that's not so much what we need. We have kind of plenty of gene for resistance, at least for most diseases. So I think it's really time that we start talking about quantitative traits, helping breeders raise that yield ceiling. That's really where the future lies to me. And for me, characterization is not enough. The reason why I'm so successful, our partnership with our gene bank, is that they actually do the crosses. I received this monococcus material 6 years ago, indicating that in this case was salinity tolerant and very, very strong DAO capacity. Great. So I thought, that's interesting. I've been spending the last 7 years waiting for one of these bloody things to give me a spike. But of course, as I have a big program and I'm doing things very quickly, I never have the time to do all the steps that are required. Last year, I gave it to the gene bank and they did the cross very quickly, very easy. So that helps. The characterization alone is not enough. And just looking at it based on Genesis, these are the 10 main gene banks having wild relative in them. If they make 20 crosses each, 40 years, we have all these crosses made to modern material. And I think that's really where the difference will come. On the other hand, I have to say the gene banks are not very well set up, at least the one I know of, for doing the field selection. Right? I think as a breeding program, we are much better designed to handle this type of thing. So I think that's where the breeder or the pre-breeder in some case, they need to come in and apply the basic breeding selection procedures, in terms of number of individuals and so forth. Right? And from my point of view, I think breeders should not be afraid of handling 20 more crosses in the program, especially if these arrive as an F2, Manelito cross that is guaranteed to be fertile. So it becomes quite easy. I had made this acronym, to say it very quickly, I call it MAGIC, the magic of wild relatives. So many crosses are made every year for elite. We need to do the same for wild relatives. Alleles are unknown in most cases from elites. So why do we need to know them in wild relatives, right? Gene bank is the best partner. For myself, the word insane, it will never happen, is the very reason why I started working in research, not with a private company. So I think there is room here to do a little bit of insane, never work activity. And then the last one is the crop breeding is in the field with simple principle. Then we need to apply them, glass house is not. With that, I think time is up. Again, I thank one more time the donors that made my life possible and the many partners and institutions that around the world contribute to my program with their germplasm and material. With that, I'll thank you. Maybe I close the sharing. Thank you.
Thank you very much, Filippo, for your candid remarks and the walk through some of the successes and some of the things that did not work in your program. I think we can go on to the questions and answers now. We have the first question from Theo van Hintem. Do you think that the diversity in durum wheat of farmers' fields worldwide has decreased or increased over the last decade? What do you expect in this? What do you expect in this respect for the future? So with regards to this, what do you expect in the future? A decrease or an increase in diversity?
Yeah, that's a tricky question to ask a breeder. Right? It's like to ask a surgeon if he should operate. Right? The global diversity in terms of land races and so forth has definitely decreased. You can see that for sure after the Green Revolution. On the other hand, the amount of diversity is captured within the varieties, I feel has remained the same. At least that's what our studies show, that diversity has not changed. So those varieties have as many alleles as before. There's a push for kind of major variety being promoted in many parts of the world, but that is also changing thanks to competition and private companies. So all in all, yes, there is a trend. There has been a major reduction in diversity some 20, 30 years ago. But since then, I think we are somewhat stable. I am hoping that some of the projects that we have submitted will go through. And then in this project, we are basically trying to bring back some of these land races, rather as in situ conservation approaches, which is fine, but linked to the whole value chain, right? We see in Italy, we have this Senatore Capelli being grown all over the country for making an organic pasta and so forth. So farmers sell that at 4 or 5 times the price of a normal crop. Works. If you just grow land races because you have no alternative, that doesn't work. But what we're trying to do is to put the land race diversity into modern material, and then hopefully try to go forward with something that has diversity within and still provides higher yields. That's my hope. But of course, I'm a breeder, so don't forget that.
This is about a question about farmers' involvement. So it's a little bit long. Let me just read it. I understand that higher yields, disease resistant, and drought tolerance are features that farmers want. I believe that is why these are a priority for breeders too. You also mentioned couscous and showed a camel enjoying the straw. Can you explain about farmers' involvement for some of us interested in seed systems? Do you think it's too late to expose farmers to selected elite material? I am asking because poor adoption is sometimes justified due to a lack of farmers' participation.
That would be a seminar, I think. Actually, I think we had quite a nice conversation around these topics in the, was it Food Forever, I think, was a conference that the Crop Trust organized with better speaker than me to talk about this. But let me say what I know. Okay. First of all, our farmers, at least the one we work with, they don't really look at the traits the same way we do. There's been, we have noticed in that their preference in their selection, they tended to prefer the second or third top yielding line, very rarely the first one, because of other things. You know, one said it's a black spike, that's the variety I was growing 20 years ago and it was great. One says, you know, I really need that straw, so I need something taller because, you know, I love, I don't know if Miguel is connected, he was saying that for barley, you know, the straw in Ethiopia actually is the roofing of the houses. So in some villages, so, you know, you cannot take away that straw to these people. So the farmers' opinions on traits, I don't think they are so well captured by breeders everywhere. Mark that live answer. Okay. So so what I was saying is that, yeah, the breeders can understand part of what farmers want, but at least in developing world, the farmer voices are not quite as loud as they should be. And there's been a huge learning process for us, at least here at Ricardo, to learn how to work with our socioeconomists, our gender specialists, yeah, we are looking at gender traits, and make that interaction. And learn better so that we can bring it back and put that into our breeding selection. Now, is it too late to do it with finished material? I think not. But I heard different opinions on that matter. At Ricardo, we only conduct participatory variety selection. We don't do participatory plant breeding because it's an issue of scale. You know, if you try to work with one village, then maybe participatory plant breeding is a great option. You know, you just go to that village, you bring a mixture of material, you work with the farm, you select it out, you help them multiply and so forth. 5 to 10 years later, you have managed to serve at least half of the farmers with a variety that is of their preference. But if you're scaling that across countries, across boundaries, across villages, it's impossible to provide that type of technical support at each farmer. So we feel that engaging them, at least when the variety is finished, we see them, appreciate them a lot. And in fact, we are using that appreciation to favor then later on the release and sale of the variety. So we have an example, this Nashit, the one I was showing, it was not purchased up until now, but we provide a letter with all the farmer requesting it, and now it will be commercially made available. So there is a back and forth. Okay, is that enough?
Yeah. What if the obviously the varietal release and all that and releasing it to farmers, it's a bit much for you to work once the variety is released. The farmer had already made his mind, so he's not going to see his characteristics that he wants in your varieties. But what if that other pathway that you showed, what if there's participatory breeding in the gene bank, the crossing with the gene bank, and the farmer is involved prior to giving you those 20 crosses more that you would put into your breed? What if the farmer is brought in that stage?
Which farmer? Because I work with 25 of them from the same country, most of them from the same village, and none of them selected the same variety. So they selected 25 different ones. So I can bring one farmer, I can bring 10, I can bring 50. It took me, my technicians that are actually people that are trained in agriculture, but they never worked in breeding before. Okay, trained agriculture, never worked in breeding before. They are born in small towns in the rural areas. It took me 5 years to train them in the selection of segregated material. Because the concept that a plot that goes up and down includes different type of genotypes is not so linear. I'm sorry, it seems simple because we have studied through it, but it's not so linear. When we work with farmers, if you have things that are very different, very, very different, they are very quick at capturing the one they want like the best. So what we are doing now is what we call farmer weighted selection. So we have many farmers that receive finished material. They not only select the one they prefer, but they declare, and that's actually published if you're interested in reading, they actually declare why they like it, which traits they like in it. Then that one becomes a weighted index where we can create mega groups of farmers, which strangely enough, they're not so geographically divided, but is really a preference. And that become a selection index that I can put in my selection, in my breeding program. So when I make that cross, I'm actually using the farmer's input to make that cross. And when I do the selection, I use the farming selection to make that input. That way, now we can integrate more than 2,000 farmers in one process.
Okay.
Again, if you are able, there are some in situ conservation sites that our gene bank intervenes into, and probably Hamed Amri would be the best one to talk about those. There, I'm sure you can do these type of things in a much more deeper scale in the fields with the people. But at our scale, I don't think we can do it. Personally, I don't think it's doable.
Right, good. We have a question from Sebastian Carpentier. Really nice talk, pleasure to listen. Fully agree with your workflow. What was not entirely clear though was how to select the 20 crosses to make from the gene bank. Surely genetic characterization, but what else do you propose that gene banks do so that the 20 crosses that you get are the ones that you're going to find successful?
I am a lucky man. I'm a very lucky man. I have Hamed Amri, I have the gene bank team here. Our selection mostly is done based on where the material was collected, or what it looks like in the field, or what it could potentially have, and is based on experience most of all. Some crosses are made for reasons that are actually characterization. So you know that you realize that some traits are of interest, but the characterization of wild relatives is for quantitative traits is really a bit pointless, and for qualitative traits, it's rare that is of interest to the breeders, unless you're really looking at those diseases for which we don't have enough material for, which are now very few. There's a couple really in my line of work that are really, so unique and so novel that we really need to find new diversity. So it's really like Hamed Amri tell me, look, this one I got it under a rock on a high plateau in Syria. I look at the grain, they look very yellow to me. I think there's something here. We discussed how difficult it is to make that cross. He said it's doable. Let's do it. Okay, this is really most of the conversation I would have. In reality, in my talk, I think I show you that the Dicocoides, we had no idea why the cross was made. Turned out they had beautiful, root depth and large grain. The Speltoides, we had no idea why we made it. Turns out that specific Speltoides and the species in general tend to have pollen that withstand high temperature much more than the others. But when the cross was made, we didn't know. Honestly, we did not know. But if you make the cross, then you find out. If you don't make the cross, you just worry about what should I cross. So my suggestion is take 20 brackets, start with those if you don't know anything more. This is my suggestion.
Interesting, very interesting. We have an influx of questions, so I'll just try and go quickly because we're almost at the point of the hour, but this is really interesting. So please stay with us beyond 9 p.m. for me or 3 p.m., 4 p.m. for you. We would like to answer all the questions. So if you have a chance, please stay. From Benjamin Killian, thank you, Filippo, for your great presentation with so many fantastic examples. Can you tell us a little bit more about your exciting participatory work in MOR? What does your work with the farmers look like? What is the most important thing? What have you learned? And what do the farmers appreciate about working with you?
Go. 1 minute. Sorry, just one comment. All the things I said about characterization is about crop wild relative. I think land races, there's much more they can be doing, including the genomic models and so forth. Sorry, just to make it clear.
Okay.
Benjamin, you were walking around and driving around with me. So you really felt how much those farmers actually tells you every second you spend with them. It basically changed my program, to be honest with you, because there are so many traits that I never even thought about. I never select for spike color in my life. When talking to them, tell you a black spike, I love it, immediately became a trait that I look for. Never before I thought about it. You know? And I don't think there are many breeders out there that select for it either. So this is the level of input that, you know, a person like that can give you. We use a lot what is called selection by stone. I think there's a beautiful article by Michael Major on Food Tank describing this process of selection by stone. I stopped there, and I would invite you to go look for that selection by stone on Google, and you see a little bit how we did it. And I think was a great process.
Wonderful. Another question from Teshome. You recommend to do crosses in gene banks. In the current formal crop development, gene banks are not mandated to do more than characterization. Are there examples of gene banks that are currently doing this?
But my own, but of course that depends on the setup, right? Yes, I know the mandate of the gene bank, and it's a very noble and already quite busy schedule to maintain. And it's not compulsory for anybody to do anything really, right? You know, breeders will continue to release varieties whether the gene bank makes those crosses or not. It's a fact. Now, if you really want to reach out, there is a step that both have to do. I think the gene bank job is to make those crosses, because simply maintaining, planting, germinating, fertilizing, this is what the gene bank knows how to do. You know, we can train somebody else to do it, but the gene bank already knows how to do it. And then the breeder have to do a step down, trying to pick that material and take responsibility to do it. If we both do that direction, this is our program. We have a beautiful floor. We are very happy. If we don't do that, we can hope that this magic pre-breeder will come and do everything. But how many institutions have the money for having an actual figure that is a pre-breeder? I don't know. Ours does not. I mean, we have a half-time person.
Yeah. Related to that, there's a question from Prasad in ICRA. Thanks for interesting talk, Filippo. Now, if gene banks also start crossing, where to draw the line between the breeding and the gene bank conservation? So where do you, I mean, you had the three very illustrative slide with the three football players. Where does the pre-breeding fall? You're advocating it to fall with the gene banks, but now it's, I think in the structure that CGIAR has, it's being done somewhere else. So where do you draw the line? Where do you suggest that this pre-breeding go? We already said that with the gene banks, but what about the current structure with the CGIAR?
Listen, we are under the current structure for a while. We have been, right? It will change again, and probably nothing will change, but it will change again. So from my point of view, when the gene bank has made an F1 or an F2 that is fertile, that, you know, was somewhat, you know, stable, segregating, but it's mostly fertile, they put that in the field, and the breeder comes in and takes it from there. Because of course my pipeline can handle that very, very easily, while for a gene bank would be really a big push out of the comfort zone to do these type of things. And honestly, sometimes the gene bank is doing that right in a glass house with a small number of plants, and the outcome cannot be the same as the numbers that we're trying. Now, that cannot touch the conservation, right? The majority of the work of the gene bank, the totality of the work of the gene bank, the mandate is the conservation and the simple characterization. The use, I hope it will become more and more common. Again, there's a hope to create this magic figure of the pre-breeder that will do everything that the other two cannot do. Private company have that type of figure. Some CG center have that type of figure. Honestly, when that figure exists, I feel that the results are worse, not better. But this is my personal feeling, meaning that the fact that comes directly into my program makes sure that everything comes out and stays there. If you create another program parallel, you need to have a very nice way of flowing it back into the other one. And that's actually, again, the three players need to find a way to coexist.
Okay. From Rebecca Labar, you raised the question whether we need more disease resistance genes. As far as I know, the ones we have already overcome or are prone to be in the future by faster evolving pathogens. Can you elaborate on what you mean by more quantitative is needed? It sounds like you refer to yield and that there is little interest in resistance genes. Can you please elaborate?
I'm not saying there is little interest in resistant genes. I think that there's a list of resistant genes that is insane. It's very, very long for at least the major rust in case of wheat. And we have not used even a quarter of them, and they're not all overcome. Huh? Most of them are still fully effective. So do we really need more rust resistant genes? Maybe. But again, if you're moving away from the boom and bust approach, but we're not putting upfront only one gene, theoretically, we should come to a point where most material is 3 or 4, and they should no longer be overcome in the future. So that frees quite a bit of investment to start looking at additional things. That said, doesn't mean that I'm not interested in disease-resistant genes. Is that simply, it wouldn't be the first decision that I make. If you give me a land race that has a disease-resistant genes, but then when I make the cross, the result is not good, I will probably drop it. You know, it's the reality of it. And yes, when I'm talking about quantitative traits, I am talking about yield. I'm talking about larger grains, root system, things like that. The most world-changing traits that wheat ever seen is the 1B, 1R translocation. And that was originally made, yes, for disease resistance in reality, but turns out that it's actually changed the root type, changed, well, okay, the quality that had to be fixed. But that trait now is so widespread, so important, and is a quantitative trait. It's a root system that we're talking about. So those are the ones we need. And if you try to characterize then genetic resources for those quantitative traits, I think the outcome will not be very useful. What you have to do is to make the cross and then characterize that cross for those traits. Then we're going to go in B&B.
Okay. Quick question about root depth. How deep should the roots go to be able to harvest an optimum yield advantage under drought? In your mind, ballpark, how deep should that be?
Here is 80 centimeters, but it really depends. If you have, depends how your soil is. In fact, in the mountains, we need shallow roots because the soil is not very deep. So it really depends on the soil type and structure and the distribution of water.
Okay. Charlotte Lusty asked the following. Thanks for the great presentation. You are persuading us about the particular value of crop oil relatives and the role of gene banks providing crop oil relatives by elite crosses. Do you think gene banks should focus on this? In other words, is it more useful than anything else that we can do, say characterization, subset, improving data, etc.?
No. I mean, subsetting, I didn't think was going to go in there, but I definitely think that everything that has to do with the collection, there's still a lot of diversity that needs to be collected. And I hope you continue doing it. Every now and then I've been able to go the gene bank to do that. And that's so bloody important. I hope it will continue forever. Maintaining all the diversity, this is really, I mean, without you guys, we'll be in a complete disaster. So these two for me remain number one. The characterization part, you know, whatever is needed for maintaining the material, yes, of course, is as important as the other two. Going beyond, personally, and I know this is a very personal view, I don't use it. I rarely use it. Maybe with land races, yes, we do this. You do it sometimes. Wild relatives, if you talk to experts that are working with wild relatives for a while, you realize that the resistant one makes you elite susceptible. The susceptible one makes you elite resistant. You never know what you're going to get out of on the other end. So for wild relative, I really, really hope. Again, land races, I think breeders can handle them, and most breeders have land races in the crossing block, and they're probably land race characterized for something that was already deemed useful. So those are okay. But for wild relative, if you could make the cross, then we can drive change together. If you don't make the cross and you expect me to do it, then I think it's going to be a stalemate.
Thank you. From Gul Ali, thanks for pointing out about useful genetic diversity versus general genetic diversity. In gene banks, genetic diversity is usually measured with markers spread over entire genomes, mostly in the non-expressed genomes. Do you think it would be useful to focus on measuring genetic diversity in the expressed genome to identify functional alleles with SNPs, for example?
I mean, really, I mean, depends on your goal. Of course, theoretically, in the expressed part of the genome, should supposedly be the most useful diversity, right? But in reality, we still know so little of the genome. I mean, what all that heterochromatin is doing there. There's a lot of transcription factor involved, a lot of, the micro-silencing system and so forth. So to me, as a breeder, what I can tell you is that looking at the diversity, I realized that the landrace with the most diversity are the one in Central Asia, okay? Which is something I would have never put in my program if I didn't realize that that's the case. Because of course, the center of origin that seems so entitling and enticing and so forth is where you have the major genes, because everything started there. So those alleles spread out. So in reality, in the center of origin, the diversity seems to be much smaller than as you, of course, you spread out and you have migration effect and you have founders effect and so forth, and the diversity creates. So from a breeding point of view, if you give me two land races and you tell me that they are very genetically diverse to each other, and I can see they are genetically diverse for my program, that's already very useful. That's a great genomic model, very simple.
We have one question, very quick question. 25 varieties selected by different farmers. What wider acceptability would each have in a wider distribution?
I'm not sure if the beginning is correct. There were not 25 varieties on 25 sites. There were 25 farms, and the variety were actually only 10. And the wider distribution, so we actually look at preferences, right? So in terms of analysis of preferences, it was not a yes or no answer. Each farmer would rank the top 4 or 5. And when you look across, yes, I mean, the top 3 tended to be kind of mostly the top 3. So yeah, so that would be also widespread. But since you're working with each farm, you try to give the top one that he wants, not the average 3 that everybody likes. So yeah, there is potential to have a one variety fits all. But of course, to make the farmer really happy, you want to give each one of them the one that they prefer. And that's what we did.
Okay. Charlotte Lusty asks, there are not many people like Ahmed Amri, and most of them are retiring. How do we try and do something to compensate for this unique knowledge disappearing?
Okay, Amri, are you there? You want to answer that one?
The ability to engage people, the ability to be able to engage people and work collectively, that too.
Yeah, I mean, yeah. Okay, I agree. What do we do next? We keep him forever. It's we put him in Svalbard. What do we want to do?
No, we clone him.
Yeah, we record him. We try to take as much time as we can with him. I know that he's training very well, the people around him within the gene bank. So he has left around himself a lot of very well trained people that work in different places. Yeah, I mean, ideally you want them to write books on this, but will they? I don't know. Yeah.
Okay, we have the last question. Most of the other ones we've been answering along the way. So I think this is time for the last one from Deeptika. I'm sorry if I didn't pronounce that right. Can you please tell me how to get purified germplasm for breeding purpose from gene banks? Because most of the samples are not, I'm sorry, this bumped up, are not, I lost my question.
I guess will be homogeneous.
Most of the samples are not homogeneous, pure enough to get parental materials. So if you get a heterozygous line or there's some segregation going on, the question is, what do you do then?
I mean, it's a whole history, and then and there's even a question. Once you have purified it, should you put it back as a new land race in the gene bank, or should you keep it out? I mean, it's a long conversation on this topic. Since most of the time I don't know what I'm crossing, why would I care? Heterozygote, homogeneous, non-homogeneous. I mean, most of the time I really don't know why that cross is going to work. So, for wheat is easy. You pick a couple of plants, whether that is a spike or something, and use those for crossing. And at the end, worst case scenario, pedigree was wrong. But I don't think farmers ever ate with pedigrees, right? It's really looking at whether or not it's the best material you can put out there. So again, immobility by indecision, by overabundance. This is what I fear. Because we cannot do everything right, we stop. No, let's do a lot of things wrong. A lot of things wrong. And maybe 20 years from now, we will say, oh, look, my great wrong things, what it looks like. So this is really my hope. This is my message.
Thank you. That was an incredibly good talk, Filippo. Thank you very much for all your candid remarks and for staying on longer. To all of you, we hope to see you next month. We have Colin Curry coming in with talking about crop genetic erosion, 100 years of evidence of what, so it will tie in very well to what you say. Filippo, what he has found with 100 years worth of evidence is a new publication that is coming out now, I think this week. So he will be discussing that publication in our next Grow webinar. Thank you very much, Filippo, again for being with us. Thank you to Faye and all the Crop Trust people for supporting us, but most of all, thank you to all of you who have come and joined and listened and participated. Stay safe, and we'll see you next time. Bye-bye.